on which we meet and acknowledge their leaders, past, present, and emerging. And today it's really a great pleasure to introduce Professor Adrian Egli, and I'd like to warmly welcome him to the meeting here. We have already met him at Peter Mac and spoken about his interests in microbiology. He's a professor of microbiology in Zurich and is visiting here for a sabbatical based at the Doherty. But we were lucky enough to have a chat with him about the problem of emerging gram-negative resistance in our patients. And of course, Adrian has been at the forefront of technology and discovery about antimicrobial resistance and host-pathogen interactions. So it's really a pleasure to have him to talk to us today. And I'll hand over to Adrian now for his presentation. Thank you, Adrian.
Yeah, thank you so much for the kind invitation and also opportunity to talk to you. I think you see my slides, I guess. So we'll talk about the evolution of AMR in immunosuppressed hosts. And what I would like to do is, first of all, give you a bit of theoretical background why there's a direct connection of immunosuppression to the risk of AMR evolution. And then I will showcase that with an example from the literature, a study which I find is extremely elegantly done. It's not from my lab, it's a study in mice. And then I will show you a study we are currently running in allogenic stem cell transplantations. And that's very early work, unpublished work I will show you. So you have a bit of everything, theory, mouse work, and then transition to humans. So first of all, if we think about the importance of the host in the host-pathogen interaction, then obviously the interactions between the immune system and the bacterium can be described from a systems biological point of view at different levels. You can start with the molecular interactions where you have two molecules interacting with each other. This results then in a biochemical signaling pathway, for example. It has consequences then on cell-cell interactions and interactions within tissues. Then you can zoom out to the whole patient to really have this systematic view of the patient. And then obviously you could also make the jump to a whole population. Now, if we look into this from the immune response, so for example, you have a cytokine, let's take interleukin-2, binding to its receptor CD25, and then you have a signal which amplifies and activates the T cell. You have an amplification of the T cell and the growth, and then that growth, for example, could result in a stimulation of B cell, and then you have humoral immunity in the patient. The antibodies are distributed throughout the body and reach different areas. And then obviously herd immunity would be the population level to describe this level or axis of the immune response. Now, when we do the same thing with bacteria, we can imagine a bacterium invading the human body. Then there's an antibiotic which is given binding to the target of the bacterium. There's a certain selection pressure which then evolves from that, and resistant bacteria would start to amplify and grow within a certain tissue. And then as soon as the bacterial amount and the corresponding immune response is so severe that it reaches a threshold for us physicians, we could then detect, for example, the patient is sick because a patient has fever, certain symptoms, etc. That would be the level of the patient. And then obviously a bacteria then can also be transmitted. So this interaction between the immune response and the microbe, it determines, as a matter of fact, the clinical course, obviously. The collateral damage, which is caused by the immune system, the inflammatory response, then generates the symptoms. So the bacteria alone, as we all know, in an immunosuppressed patient, sometimes the symptoms can be very weak. So it's really the immune response which tells us usually if a patient is sick or not. And then the immune control is very important to reduce the bacterial burden. Source control happens inside the body, and there are less bacteria. So the immune system is extremely important in this interaction, and what we try to do if there's an immunosuppressed host present, we try to compensate with antibiotics. And what we do here is that we try to expose the patient very rapidly to an effective antibiotic treatment. And this is very nicely shown here in this, oldie but goldie, I would say, as a paper where you can see the time it takes for an effective antibiotic in hypotension, so that's septic shock here, and you see how the survival fraction actually goes down over time. So it's very important that we administer an effective antibiotic in an immunosuppressed patient. So if we look into this interaction between bacterial growth and the antibiotic concentration more systematically, we can obviously describe that in certain statistical models. What you can see here is concentration over time of exposure. So here you have a time axis. That's the concentration of the drug. The dashed line here is the minimal inhibitory concentration. So it's very important to expose a bacterium over a long time above the minimal inhibitory concentration. So this is time above MIC. And then you can obviously also say how much more was the concentration of the antibiotic above this MIC. So these are two important values when you generate mathematical models. You can also integrate into this the bacterial growth rate. And this is here a Hill function. I will not go here into the mathematical details, but what is very important here, you can see on the y-axis the bacterial growth. So this can be described from the maximum. The bacteria can go to the minimum where the bacteria is basically dead. And you can see here that along a line of MIC, so that's the amount of antibiotics you give above a certain MIC concentration, you can see that the bacterial growth obviously is inhibited stronger. You can further add here modifications with the kappa function you see here. So a stronger antibiotic, for example, would result in a steeper decline of the growth, where something which is weaker would reduce this steepness, and then you have a bit more a flat reduction of the growth curve. And finally, you can also measure this in the lab. You can do so-called time kill curves, where you have the bacterial density plotted here on the y-axis, and you measure over time how much bacteria die. And you see here different antibiotic drug concentrations, in this case ciprofloxacin. So if you give 2 micrograms per milliliter, you see here a very steep decline, and when you have lower concentration, the decline is not as steep. So the higher the concentration, the lower the time to reduce bacterial density. And this is very, very important to reduce the bacterial density, and I will explain to you why this is so important. So this hit hard concept is critical. Because what influences the MIC? I mean, there are clearly factors from the bacterium itself, so I will not talk here directly about the specific resistance mechanisms, but there's a second important aspect, and this is the inoculum effect. And the inoculum effect is a laboratory phenomenon that describes that is described as a significant increase in the minimal inhibitory concentration of an antibiotic when the number of organisms inoculated is increased. Now, this is a phenomenon we try to compensate in the lab. As you all, or some of you might know, in the lab, we generate a so-called McFarland concentration. So this is a certain density of bacteria, and we try to standardize that and always use the same concentration. And this is in these two examples here. I have one example with meropenem and one with cefepime. You see here this dashed line. That's actually what is done in the lab. Now, if you decrease your inoculum, you will have less bacteria, and then there's a reduction in the log change of the MIC. So the bacteria become more susceptible to a certain concentration of antibiotics because they are exposed to a higher per se concentration of the drug. And the opposite is true if you increase the amount of bacteria. If your inoculum gets larger, then you can see that the MIC relatively increases, and you need to give more antibiotic drug to actually kill those bacteria. And that's very important if you think about an abscess, for example, where you clearly have in the human body higher concentrations, whereas there are certain, let's say, urinary tract infections where you might have lower concentrations of bacteria. And this is in the lab to a large degree ignored, I have to say. We are not generating, for example, if we know this comes from an abscess, we would not increase our inoculum and then report a more realistic MIC value here. Now, what contributes to the inoculum? Obviously, the inflammatory response, because if you have a strong immune response, your bacteria are under a certain stress, they are cleared from the system, and your inoculum is also reduced. And you can see this here schematically on this graph. On the left side, the bacteria stimulates pattern recognition receptors, so toll-like receptors, for example. And then within hours, you have from the innate immune response a certain inflammatory signal. And then after a few days, then obviously the adaptive immune system kicks in and then has also a control, obviously, over the bacteria. So the role of the immune system during infection is to, on one hand, control the growth of the bacteria, and then obviously also immediately remove invasive bacteria, which, bypass, for example, the epithelial layers. So a reduced growth results in a smaller inoculum of the bacteria, and so therefore it's also easier to treat a patient in this setting. Now, if the immune system is suppressed, then you have an increased risk for invasive infections because there's a higher exposure to the antibiotics. That's certainly clearly the case that when you have more exposure to antibiotics, there's also a selection pressure on the bacteria, but more bacteria during infection also happens, so there's a lack of control and a reduced clearing due to the immune system. Now, the inoculum is critical for the evolution of resistance because if the inoculum is big, then it's more likely that the bacteria can generate mutations because you have more cells under a certain selection bias, and therefore mutations will accumulate. You're going to see this actually then later on in this mouse paper, which I mentioned in the beginning. And then on the other hand, you also have more targets for the antibiotics because there's more biomass and there's a certain amount of drug targets, so there's a relative reduction in the concentration. So the immunodeficiency shifts the infection risks in bacterial populations through, first of all, direct fitness effects and through the impact on the antibiotic clearance. So the fitness landscape, which you can see here down on the graph, encountered by a bacterial pathogen greatly varies throughout patient population in terms of their host immunity. And so a severely immunocompromised host, which is shown here, favors an environment for antibiotic resistance due to the lack of the immune-mediated competition and increased opportunities to evolve. So you can see here the relative bacterial fitness in an immunocompromised host. It gets stronger in the case of resistant bacteria because there's this second hit of the immune system is lacking. And here you can see the immune-mediated bottleneck, which is wider in the case of an immunocompromised host to a healthy immunocompetent host where it's much more narrow. If we now bring this introduction to an end and kind of finish this theoretical part of the talk, again, I would just like to highlight here the most important aspect. So you have growth rate on the y-axis and the antibiotic concentration on the x-axis. And you see here a susceptible strain, how it goes down when it reaches the minimal inhibitory concentrations. That's the definition. Then there's no longer growth. But before you actually reach the MIC, you have here this window of opportunity, which is defined by the MSC. So that's the minimal selective concentration. So there's a selection and window of opportunity for the bacteria to accumulate mutations. And if you go very high with your drug concentrations, you will eventually reach the MPC, which is the mutant preventive concentration. At that time, you also reduce the growth of resistant subpopulations, and then there's basically no longer growth. So if we summarize that, in a patient with immunosuppression, you will have, due to the higher inoculum effect and the lack of this immune control, you have a higher risk of the bacteria that they can collect mutations down the road and resistant strains are selected. So for this reason, we should try to hit hard and fast if possible and go for higher concentrations if this can be done in terms of toxicity profiles. So that was quite theoretical, but I would like now to tell you this, how this actually also works in a mouse model. And there's a beautiful paper which was published about 2 years ago from Hua and Bush. And what they did is they set up a mouse model to explore this with the case of Acinetobacter baumannii. So certainly a bacterium which is extremely relevant in immunosuppressed patients. And what you can see here from the experimental setup, what they did is they had 2 mice groups, immunodeficient mice and immunocompetent mice. And they treated them here, they inoculated them with 10 to the 8 colony-forming units of Acinetobacter baumannii, and then they treated the 2 mice with ciprofloxacin, and after some time, they homogenized the lungs and plated the lung material on LB agar plates and quantified the lung bacterial loads. And what you can see here is that the immunosuppressed patients, they had a relatively stable concentration over different passages. They did of around 10 to the 5 colony-forming units, whereas in the immunosuppressed patients, there was a clear increase, and this increase even was then larger than the actual inoculum size. Now, how can this be explained? One key factor is exactly this selection of drug-resistant strains. So you see here 3 different lineages of mice. So these are 3 immunocompetent mice populations. And you see here the fraction of bacteria in percent, which had a certain resistance level. So for example, the white ones, they were fully susceptible to ciprofloxacin. The pink ones had an MIC of 0.25, and the green colonies here had an MIC of 1. Overall, when you look over these different passages, this stayed the same. So there was no accumulation, let's say, of certain populations. Here you can see mutational frequencies. They indeed observed some mutations. So that's passages on a agar plate. And it seems that there were some accumulations of mutations, which could be a plate adaptation, basically. But now what is striking is what happens in the immunosuppressed mice, because this is exactly what I told you before. There's an accumulation of resistant subpopulations, and they receive the exact same antibiotic treatment. These were the same mice lines. So the only difference is that these mice were immunosuppressed and the mice before not. And what you can then see here, there was an accumulation of certain point mutations, and these point mutations were resulting in an overexpression of an efflux pump. And you can see here now the consequence of this. This is exactly, again, these time kill curves where you have the antibiotic, given over a certain time period. And you can see here the colony forming units. And you see that some of the strains which accumulated certain types of mutations, they were cleared less effectively than other bacteria. So I think that's a very, very elegant paper who explored this impact of the immune system on the selective pressure of the antibiotics. So to summarize this first part, host-pathogen interaction is complex, obviously. The immune response is absolutely critical to control bacterial growth, and a reduced control of this bacterial growth results in a higher inoculum. And the higher inoculum is an opportunity for the bacteria to accumulate certain mutations, which then lead to resistance. Okay. Now I would like to switch to our study, which we have started about a year ago, and it's really a lot of preliminary work. And I just want to kind of, also trigger a discussion, obviously. And if there's enough time later on, or maybe if you have ideas, please also drop me an email after the talk. So what we try to do here is we set up a stem cell transplant cohort, which I will introduce to you in a second, and we follow up these patients over time. So we have a lot of clinical information, access to all the electronic healthcare records, so we know how much antibiotic is given, what kind of immunosuppression these patients have, also during the neutropenic phase, obviously, and then the immune reconstitution is monitored very tightly. And we get a stool sample every week. So we have a lot of stool samples you will see in a second, and we plate out these bacteria and look into the antibiotic distribution. So there are three leading questions we have. First of all, what are the risk factors influencing the evolution of antibiotic microbial resistance within the host? What is the role of the host response? So we want to look at this in organoid models, and that's very early work. I mean, my lab has not established an organoid model, so here we will clearly also collaborate with colleagues, but we want to first of all look at the epithelial interactions and then make these models more and more complex by adding, if possible, inflammatory stimuli or immune cells. And then the third question is, what is the contribution of other bacteria? Because obviously in the gut, you have a lot of other bacteria. In abscesses, very often you also have more than one species. So the interaction with the bacterial community is an important question for us. So here is the study design, and this is done together with colleagues from the University Hospital in Basel. This is about an hour from the University of Zurich away. So everything is 1 hour away in Switzerland, I have to say. So the idea is here to collect samples already before the allogeneic stem cell transplantation. We collect serum, stool, skin swabs, and skin biopsies. So we will also answer other questions. So for example, what are certain bacteria on the skin doing in terms of GVHD risks? That's another question we have. But for this particular talk now, I will focus on the AMR aspect. We have clinical assessment at each time points, which is documented in the red cap, and we know exactly all sorts of drug exposures, not just antibiotics. And you can see here on the timeline, we have a preconditioning period, then the actual transplantation, there's pre-engraftment, and there's post-conditioning, the late phase, and then we have quite long-term follow-ups. You see here the serum samples we take, so in the beginning every week and then every 2 weeks. Stool samples we get every week. Skin swabs, we have a bit larger gaps, so 1 month, 3 months, and then 6 months. And then we also have an initial biopsy, and we have dynamic sampling at the skin. So if there's a specific site which develops graft versus host disease, there's an extra swab taking from that site and from the healthy counter site, if that exists. And obviously all patients, this study follows the ethical protocols. They're all asked and consented, and they can also decide what they want to do, if they want to donate stool and skin swabs, or if just one or the other. And a large fraction, I would say 70% of the patients actually give both materials. So here you can see the recruitment. So our recruitment goal is to recruit 100 patients. Currently, we are at patient 83, and you can see that in about 60, 70 patients, we already have the very initial period, which is probably the most interesting part of the study, is already finished. So this first half a year of follow-up. And we will close the study end of April. So most likely we will not reach 100 allogenic stem cell transplantation. Perhaps we will reach 90, but that's still absolutely plenty. We collect two types of stool, one which contains some preservatives, glycerol, etc. And we also do metagenomic or 16S based analysis, not shotgun at the moment. Eventually we will do it, but that's a bit of a funding problem here. And then what we also collect is native stools, and there we do metabolomic profiling from the stool samples. And the stools are processed in the following way. We weight them so that we have also the relative concentration of the stool. We know that. Then we do aliquots, where we have a very, very nice biobank information management system, a BIMS. So the whole biobank is well organized. And then we extract already DNA from these samples for 16S sequencing, and that's ongoing work. We also then plate the stool samples. So we have two plating strategies. We do aerobic plating. So that's what the lab technicians are doing, and anaerobic plating. You can see here the plate types, so blood agar plates, brucella plates, and so on. And then it's incubated in the aerobic concentration overnight, but in the anaerobic conditions 2 to 5 days. And then when we see colony growth, we go with a loop and collect like a full range of bacteria. So there we don't pick individual colonies. The PhD students, which you will later on see, they have additional plating strategies which come on top of that. But that's just the harvesting of the material and the samples we actually put together. Then from the serum, we also collect serum. We do aliquots as well. One aliquot is sent to ETH Zurich for metabolic profiling. And then we have 4 additional aliquots for future studies. So we could look, for example, into certain biomarkers, cytokine profiles as an example, or cell-free DNA assays could also potentially be done. Then the skin sampling, this I will keep short because it's not part of this particular study. There we collect at 5 different locations, a buccal swab, and then we have typical locations where graft versus host disease appears, and we sample those. And here we also do a direct extraction of the DNA. The leftover material is also stored, and here we already started to analyze the 16S sequencing data of these skin locations. And then we also have a biopsy. Here we are still optimizing some of our DNA extraction, but a first trial of a skin biopsy was actually successful, so we can detect bacteria. Good. The amount of samples is ridiculous. We generate in this study. You can imagine we have so far almost 7,000 samples collected. So here you see the biopsies. You have here then the skin swabs, the serum samples, and the stool samples which are collected. So and then obviously when we plate them out, there's additional subsamples generated, and you will later on see what the students do. They then even start to pick individual colonies. So that's a a cohort, I can tell you, which will keep my lab busy probably over the next 10 years. And many PhD students will actually work on that. But now we collect all of this. So what? So 2 PhD students are currently working. This is a study financed by the Swiss National Science Foundation. The first PhD student works mainly on the evolution of AMR over time. So what kind of mutations accumulate and how does the heterogeneity of individual bacteria look like over time. And the second student mainly looks into the interaction with the bacterial communities. So let me first start with the first PhD student. This is Elisa, a very talented PhD student. She has started here with this work and really tries to answer the questions, what is genetically happening in these cohorts. So from sensitive to resistant, and obviously in this period where a lot of antibiotics are given, for example, due to neutropenic fever, and then there's a direct exposure to antibiotics. What she does is she takes stool samples, she streaks them out on 2 different plates. We have a colony picking robot which can pick colonies automatically, and this colony picking robot picks the colonies, loads automatically a MALDI-TOF target. So we know then, and I have an own MALDI-TOF in my lab, where we can identify what kind of species there are. So especially from COS, this is important. From McConkey, obviously, you will have a lot of E. coli. But then it goes back to the exact same position and picks another sample, and thereby we build up a biobank of individual bacterial cells. And what you can see here is really results from the first couple of patients. Although it looks like a 16S plot, this is actually phenotypically what is growing in about 20 colonies. So you can see here, each column is a patient. Each row is a specific type of agar plate. And you can see here the different colors indicate various bacteria. So the pink one, that's E. coli, that's pink on the McConkey, so it's pink here. Then we have Enterococcus fecalis, which is orange. We have Enterococcus faecium, which is red. And in blue, you see Enterococcus avium. Now, what is quite interesting is that if you look at the different patients, there is a bit of heterogeneity. I mean, these 4 species, which I mentioned, are clearly dominating, but sometimes you have interesting switches. So some patients don't have a lot of E. coli and are not so interesting for us to follow up now, because Elisa will focus on the evolution of E. coli. And then you see quite a bit of a dynamic, when is E. coli coming, when is it disappearing? And she will now work this up, and obviously we will also have matching 16S data for that. But all the colonies we pick, we freeze. So if somebody is interested in, let's say, Enterococcus faecium evolution, please reach out to me. I'm more a gram-negative person. What she then also does is she takes then these E. coli strains and streaks them out again, generates a McFarland and measures by disk diffusion inhibition zones of 6 critical antibiotics, which are commonly given in these patients. So amikacin, levofloxacin, piptazo, cefepime, and 2 more beta-lactam antibiotics. And these plates are read by a so-called SIR-scan system. That's an automated plate reader which can process these plates. And what she has now done so far is measured 228 inhibition zones of 38 E. colis. And here is just a snapshot of one patient. So obviously per time point, we have about 10 to 20 E. coli strains. And you see here the diversity at each time point. So this is time point 18, time point 19, time point 21, and so on. So from left to right, you see the different time points. You have here on the y-axis, you see the inhibition zone. So the higher the inhibition zone, the more susceptible the strain is. The lower it goes, the more resistant it is. And you can see that for certain antibiotics, it's really boring because nothing happens in this particular patient, which is is interesting. Sometimes it might even get a bit more susceptible at later time points. But what is also interesting is that we have sometimes different subpopulations inside the same patient. And here in this case with levofloxacin, you see a clear increase of resistance. And so that's just one patient. She has now started to work this up in more detail. So the next steps here are to obviously look into this. I think a very, very important challenge we currently face is how to sequence this. And I just recently talked to a colleague here locally where a sweep shotgun metagenomic approach might be a possibility. So here I would be totally interested to kind of follow this technology up and see if this would be a way how we could separate individual E. coli strains from a sweep collection of from these plates. And until end of the year, I hope that she can process 1 to 2 patients per week. And then we have, towards the end of the year, this cohort worked up. The second student, Mariella, is looking more into the host, the community aspects of the bacteria. So if you give an antibiotic and there's a collection of different bacteria, how do they influence each other? And are there, for example, certain bacteria which protect each other in certain ways? And this is the topic she's looking into. And to do that, we have thought about 3 main parts of her project. She really just started a couple of months ago. The first one is here that we will generate dual plates where we have, and we have these plates now in the lab, where you can grow bacteria in one plate and have a second bacteria in the other plate. And what happens is these bacteria are not in direct contact, but the metabolites affect each other. So we can, for example, study if there's an E. coli and there's a partner bacteria on the other side, is the impact of the antibiotic the same if it's alone or if there are other bacteria present? And she will test now 5 different pathogens and 10 commensals in anaerobic conditions. So that's reflecting better what is going on in the gut. And then she will take this data and bring this in a Lotka-Volterra model to simulate the effect. The growth data will then serve as a parameter in this model, and we will simulate the temporal changes of the species abundance within a community. And then in the last step, she will do a transwell plate. That's then our first step towards organoids, where we plate epithelial cells. Most likely we will not take primary cells from patients. Obviously, we anyways don't have gut biopsies from these patients, but a cell line, and then add bacteria on top of this and study this. So here we still have to figure out how to best simulate this in the different aerobic conditions, where you have the lumen of the patient would be anaerobic, and then the tissue is more oxygenized. So yeah, we have to see how to best do that. And here we are certainly also looking for collaboration partners in this PhD. So here the steps would be to study these interactions in more detail. So for the interest of time, I will go here not too much into the details. So I think in step 2, I've shown you that we are processing samples of a very large study. We have a unique opportunity to study the AMR evolution in the context of immunosuppression. We have a massive collection of isolates available and complex logistics we measure, we have in the lab. So there are 4 lab technicians at the moment just processing these samples. So it's really a lot of money and time effort which goes into this study. We have a fairly heterogeneous situation with patient-to-patient unique profiles of what are the dominating species. At the moment, still too little data on phenotypes, but this will obviously come. And we will integrate at the end of the project different data layers. So we have on one hand all these phenotypic informations, as you have seen. Then we have the 16S data, the metabolic profile data, which comes from ETH. And this project is also done in a larger consortium system for the metabolic profiling, for example. Yeah, and I think to kind of sum up this talk, so AMR evolution is dependent on exposure to drug. The host immune response plays an extremely important role there, and I think clinically this is not appreciated enough. Immunosuppressed patients offer a ground to study the evolution of AMR, and there's an opportunity to study, obviously, bacterial evolution in these patients. Nevertheless, studies are complex due to very high patient-to-patient heterogeneity, and I hope that we can describe certain generalizable patterns in these 100 patients, and I'm very, very open for collaborations if you have ideas. And this is my lab, which I want to thank all the lab members contributing to this work, and obviously all the funding sources we have. And I thank you very much for your attention. I hope we are still on time. Thank you.
Very soon.
Thank you so much, Adrian. That's a really fascinating discussion about antimicrobial resistance. And I think we're going to have some time for questions. We did have a case presentation scheduled, but I think we'll hold that over till next week so that we can actually spend some time discussing this data and talking about what you've shown us today, which is really fascinating. I might kick off with a question initially, and I just wondered, and maybe it's something one of your PhDs will be looking at, is this evolution of resistance dependent upon the pathogen itself? Can you actually see more tendency to resistance in one pathogen than another? And my second question was about, are you looking at fungal species at all in this study?
Yeah, thank you very much for these important questions. So the PhD student Elisa is mainly focusing on E. coli. So because, picking at the end, she will explore thousands of individual E. coli strains in these patients over time. So she's just focusing on E. coli. I have potentially a second PhD student from Indonesia joining in summer, and she would be very interested in certain anaerobic bacteria. So we will also look into anaerobes. It is very likely that the mutational capacity, and then also depends obviously on the drugs which are given, etc., are different from species to species. I think that's we will certainly look into this. Other abundant gram-negative bacteria we were interested would be Enterobacter cloacae, for example, or Klebsiella. That would be very, very interesting. Luckily, the baseline resistance rate in this population in Switzerland is very low. So the colonization rate of ESBL E. coli, for example, in the Swiss population is 2% only. And in maybe this patient cohort, which is a bit more exposed to hospital visits, it's maybe 3 to 4%, but we will not have a high baseline, and that actually allows us to study these effects. Now, we're currently not looking into fungi. We have stored perfectly all the stool samples, so if somebody is interested in yeast, for example, we could certainly look into this, yeah.
Really fascinating, Adrian. Can you tell me, are you able to give us and I popped on a couple of minutes late, so I don't know if you talked about it, but what is your current neutropenic fever protocol? Do you use quinine prophylaxis? What's your rate of inappropriate use in the community? We know the patterns of use in Switzerland are quite different to Australia, but it is interesting for us to hear what your, for example, neutropenic fever protocol would be.
Yeah, so there are two cycles they give in neutropenic fever. And it's always a broad-spectrum beta-lactamase antibiotic with an aminoglycoside. So that's given, and then if there's a new fever episode, for example, you would switch to another antibiotic combination. So it's a dual combination, which is given the aminoglycoside for 3 days initially, and then that is reduced. there's no prophylaxis with quinolones, but I can clearly tell you, at least when I was a resident on that ward where the study was done, this is also the reason I still knew the head physician there very well, and I was his student, let's say, as a young doctor. I realized that there was a lot of levofloxacin given in the patient. So especially right after, when they were released from the isolation ward, I mean, they were clearly still severely immunosuppressed, and as soon as they had some sort of respiratory symptoms, levofloxacin was given to them. So it was not a constant exposure, but clearly a high exposure for sure.
There's also been a lot of chitter-chatter around Cefepime versus Pip/Tas exposure to anaerobes on the microbiome. And there was all those individual patient level meta-analysis that still seem to show a signal for Cefepime for patient mortality. So, I'm at stewardship end, right? Interesting what we do about all this at the other end. So have you got any thoughts about any of that evidence?
No, it's a very good input. Thanks a lot. We can certainly look into this, especially because we have the 6NS data. So the microbiome effects we can also look into, not just phenotypically, but also genomically, obviously then also with a deeper resolution. And all the antibiotic drug exposure is available for us, so we can also look into this. If we see a pattern with only 100 patients, I'm not sure if we are powered enough, but it's a good input to actually look into. It's not our main question, obviously, but
it's a really big discussion point, and you may not be aware, but we've just finalised a six-part Australia-New Zealand consensus guideline for neutropenic fever management, which will include empiric guidelines, how to manage persistent recurrent fever, role of prophylaxis, antimicrobial stewardship and infection prevention, principles and practices around identifying patients at risk, and then there's a consumer-facing chapter. And so in Australia, we haven't used dual antimicrobials for 10 years, actually, only for those patients in shock. So routine 3 days of venetoclax would never ordinarily be done in Australia for quite some time now. So I'm interested to hear that you're still doing it in Switzerland.
Yeah, I mean, as said, I'm happy to actually look into it.
Yeah.
What is probably now good is when I look at the antibiotics we test, there's Cefepime on it, Pip/Tas is on it. Initially, I thought, should we add also a carbapenem? But carbapenem resistance obviously is very, very low in our population. So, yeah.
But they do use it as an anxiolytic, so it's often that this next step. So even though it might not be resistance, I still maybe at a 16S level it might be really interesting to see what happens. I don't know, just as a secondary question.
Thanks a lot for the input.
You're on mute.
Liv, go ahead, please.
Thanks, Monica. Thanks, Adrian. My name's Liv. I'm one of the ID physicians at one of the Melbourne hospitals and Monica's PhD student, and I'm dabbling in the microbiome a little bit for my PhD. So absolutely blown away by and I'm sorry I missed the start of your talk because I was on a ward round, but the breadth and depth and sheer magnitude of the sampling and processing that you're doing is it's a bit crazy, it's mind blowing and inspirational actually. And I guess I was interested particularly, we've tried to collect some anaerobic samples or samples fresh to process anaerobically in the lab, and as a sole PhD doing this, it's been challenging because patients don't generally open their bowels on command and provide samples in hours. So it's a lot of work for a PhD student to be managing. I was interested if I might ask about the sheer number of people you have in your lab operating, and is this a 24/7, 365-day process that you currently have operating, and do you have all the ward staff involved? And it just sounds huge.
Yeah, no, the way how we do that is the patients actually, as you say, the bowel cannot be emptied on command. So what they have is they have a collection sample at home, and they are instructed to immediately freeze their stool sample. So they have special boxes they can actually freeze at home. And then we provide them with camping freezers. So typical Aussie, I would say, camping is popular here. So we have portable freezers, -20 freezers they can carry around, and they can bring the sample frozen to the hematological ward, because in the beginning, after they are released from the ward, they anyways, in the beginning, they even have a daily visit and then weekly visit every 2 weeks. And so we then collect these samples. They are then transferred to a -80 in the hematological ward. And my team goes twice a week by, also with a portable camping freezer. So we really make sure that the integrity of the sample is maintained. So on this study, currently there are 7 people working. We have 2 PhD students, 1 PhD student as a master student, and 4 lab technicians who help with the processing, etc. It really takes a lot of resources. So I'm also happy when this study actually goes a bit to an end in terms of the recruitment, you can imagine. But still, there's still this follow-up period. But we are at the moment, we are really at the peak phase of the samples coming in, and everybody is busy in the lab. And it's not that we, I mean, the sample are processed not on the weekend, for example. It's really from Monday to Friday, but it really keeps the amount of people busy. Yeah.
Very civilized. May I ask one more question, if I may, out of interest? I was surprised when we were, I guess, approaching patients to participate in this type of work, how much community understanding or appreciation for the microbiome and willingness to participate in this kind of, like, quite enthusiastic participation from people who you might not think would want to put their own poop in a tube for research. Do you find a similar sort of thing in your cohort?
Yes, I would say. I mean, the thing is with, that's at least my experience with either solid organ transplant patients or hematological stem cell transplant patients. They're very altruistic. They know, how. They're also interested in new discoveries and contributing basically to it. In this particular study, indeed, it was interesting. Some people didn't want it a skin swab, and others didn't want to give a stool sample, which was. But then one or the other sample was okay, but the majority, as I mentioned, about 70% actually give both sample types. So I would say the overall response rate is really good. I think now what I heard from, that was just, by the way, the 7 people is just from my lab. On the hematological ward, I could show you a group picture. There were like 10 people also involved in helping to recruit the patients. They have dedicated study nurses, which approve, consent. And I mean, just in terms of consenting, you have lots to read and explain the study to them. And I think because we have dedicated personal recruiting, we are also quite successful. So the overall inclusion rate is more than 80% of the patients who ask actually participate.
Amazing. Thank you. I think you're on mute, Monica.
That's impressive to get that kind of consent as well. So there must be a very good team that you have working with you on the wards. Yeah.
Yeah, we did in the past an influenza vaccination study in exactly the same ward, and there it was also even 90% actually. But that was a vaccination which they anyway received, and then they came several times to donate immune cells. A stool sample clearly is a bit more cumbersome, but people are generally very interested at the moment in the topic of the microbiome, I would say. It's a bit a hype theme.
So, well, thank you, Adrian. I don't think we have any more questions, so we may finish a few minutes early, but I wanted to thank you again for making the time to present to us. It's been very stimulating. You've given us a lot of food for thought, and I think we'd be very interested in exploring some potential collaborations with your group. I think we'll need to go away and think and talk as a group about how we might how we might do that and what we might look at. But you're with us in Melbourne for a little bit longer. Is that correct, Adrian?
Yes, exactly, until end of next week. But then I'm also, please reach out to me if you want to have a coffee with me or talk with me. I'm very happy to come. And then in September I will be back already. There's a epidemiological sequencing conference here in Melbourne. So first week of September I'm also back.
Okay, thank you. I'm sure we'll reach out to you. And it's been wonderful having you talk today. And thank you very much for your time and for a great lecture. Thank you.
Thank you very much. Have a wonderful day. Bye-bye.
Yeah. Bye-bye.